Brilliant. Okay, then, well, hi everyone, and welcome to the 14th TIA Centre seminar of the academic year. So those that don't know me, my name is Adam Shepherd. I'm a postdoc here at TIA Centre. And for those people that are new to our seminars, we aim to invite researchers from across the globe to present new and exciting work. But before we get started today, I'd just like to remind everyone that a few of us members from the TIA Centre, so Nasir, Musfa, Nida and myself, are hosting a special session at the MIUA 2024 conference in Manchester titled Recent Advances in Computational Pathology. And the full paper deadline has been extended to the 15th of April. So it's a really good opportunity if anyone wants to submit their work for presentation, and it will be published in Springer. So yeah, and if you have any questions, feel free to get in touch with me. But moving on to seminar, I would like to introduce Dr. Mohammed Shaban today. So we're especially excited to have Shaban join us today, as he's of course an alumni of the TIA Centre. Shaban is a machine learning scientist at the AI for Pathology Image Analysis Lab, associated with Harvard Med School. This role follows his completion of over 2 years of postdoc research at the same lab. He earned his PhD in computer science from the University of Warwick, focusing on medical image analysis. He has over 8 years of research experience in computer vision, deep learning, and AI-driven medical image analysis. His primary focus is developing advanced algorithms for critical clinical applications in computational pathology. And the title of Shaban's talk today is MAPS, a pathologist-level cell type annotation from tissue images through machine learning. So brilliant. Thank you very much for joining us today, Shaban. And with that, if you'd like to get started, then brilliant.
Okay, thank you for your kind introduction. Yeah, so it's like always a pleasure to present at where you have like done your PhD. Like I spent 4 years over there. So yeah, that's great. Okay, so I can like start sharing my screen. So, let's see. Okay, so can you see my screen?
Yeah, we can see your screen. Okay. Do you want to go into presenter view?
Oh yeah. So I think okay. So now
we can see that. Okay. Yeah.
Okay, that's good. Right. Okay, so thank you everyone for joining for this this talk. So this talk is basically based on one of my publication which I recently published, like very first day of this year in Nature Communication, and so that this title is a little bit like a revised title of the paper. So it's like basically on cell phenotyping in highly multiplex images through machine learning, right? So in this talk, I will like first I will like briefly define these terms, and then I'll like state the problem that I have addressed in this publication, and then also give like one slide overview of existing works, followed by detailed sort of description of results, datasets, and the proposed model. And at the end, I have like few future directions that I would like to discuss. So and also get like kind of your feedback on those directions, right? So the first thing first, so the first question is like, what is cell phenotyping? So in like medical term, it could be like cell phenotyping is the process of characterization and quantification of complex molecular interactions within cells, right? But in technical sense, or like in machine learning sense, it's basically a classification problem where we want to classify cells into different cell types. So and there are like many cell phenotypes. It depends like for a specific tissue. So and it depends on the pathologist. Let's say for a specific project, pathologist or whoever is designing the project, they may have like a selective number of cell phenotypes, but there are like common one are like T cells, B cells, and then macrophages, and then there are also like subtypes as well, like M1 macrophages, M2 macrophages. Like there are so many. It depends like how many someone want to define for a given tissue type or cancer type. Right? So now the next thing is like why cell phenotyping is important. So one of it's basically a key step to understand the tissue microenvironment or tumor microenvironment of a given tissue, and with with this, we can also like understand better the disease model, and also we can discover and then evaluate the drugs for that specific disease model. And it's also like useful for like if we want to find something new insights in a tumor microenvironment. So self-phenotyping is one of the key steps where we first define like what are the different types of cells, and then we do like some sort of like spatial analysis, or there we try to like understand or analyze their spatial interaction. So now the question is like, now we know that like what is self-phenotyping and why it's important. Now the question is like, can we do self-phenotyping using H&E images that we usually work, especially in like at Warwick, like the most of the work is basically on H&E images, especially when I was there. So, and the other thing is like H&E stain is basically the most widely used stain for histology-based medical diagnosis. So which means we will have lots of samples for different cancer types and different tissue types. So it makes sense if we can do self-phenotyping using H&E images. But and then the other thing is like with H&E images, we basically the H&E stain basically highlight the nuclear marker and the cytoplasmic area of the nuclear. So with this stain, we can see the overall structure of the tissue and also where are the different cells, right? So, and like other question is like what type of cells we can see. So basically in H&E images, we can see like epithelial cells, but the issue is like we cannot really like it's really hard to actually classify those cells into different subtypes. Let's say we can, like a pathologist can easily say that like, okay, these there are like immune cells, but it's really hard for them to tell like whether they are T cells, B cells. Like in some cases, it's obvious depending on the like the tissue type and the area of the tissue, but usually it's hard to call all like all the subtypes of each immune cell, right? So then obviously if we cannot do this thing with H&E images, then we need something else. So that is basically multiplex imaging. So multiplex images is basically kind of like simultaneous visualization of multiple markers or molecular targets within the same tissue sample. So we can still say H&E image as a multiplex image where there are only like two plex. One is like nuclear marker, another one is like cytoplasmic marker. But for detailed cell phenotyping, we really need like way more than two markers, right? So for that, there are like several technologies which support or like up to like 60 markers. So that's kind of way to go for detailed cell phenotyping, right? So here you can see an example where you can see an H&E image where you can perhaps see like cells and cytoplasmic area and overall structure of the tissue. But then in the overlay, you see like the cell, like different colors where each color represent one specific marker. So perhaps here you can also see like one of the color is representing like cell boundary, which is kind of cell membrane, and then one is like a nuclear marker, and then the green one is perhaps the cytoplasmic marker. So basically that's the benefit of this like this new technology where we can have like one specific marker for a specific cell type, so which is really great. So and then the common like CODEX, maybe SciSafe and CellDive are like few common technologies, but there are few others as well. So in this talk, I will only give a like really short overview of like CODEX and maybe multiplex imaging technologies. So both like CODEX for like 40 plus markers, and it basically process the image or the tissue in an iterative manner. And in each iteration, it considers like three antibodies, which are basically associated with specific markers, and then one nuclear marker. And in each cycle, we get like 4 channels. And so, and then the nuclear marker is basically used to align all those markers in different channels, right? So, but at the end, depending on the number of markers, we will get an image of, let's say, height multiplied by width multiplied by number of channels. So if there are like 40 channels, so then we have like 40 channels, right? So that's kind of a technical difference. Like in HID images, we usually have like RGB colors, like only 3 channels, but in multiplex imaging, the number of channels are basically equal to the number of markers we have. So the next technology is like MIBI. The CODEX is autofluorescence based technology, and MIBI is like it's like multiplex ion beam imaging, which is based which use secondary ion mass spectrometry. Like the overall working of this technology is a bit complex, but just kind of for the overview, it again produce the same type of image where we have like n number of channels, right? So now the question is like with these technologies we will get the like different markers, maybe like 10, 20 or up to 60, right? So how we actually get the phenotypes? So basically for each marker we get some cell response, which basically highlight the structural characteristics of that cell, right? And then pathologists define the phenotypes. Like they basically based on their knowledge, they define like based on the combination of different markers, they define like, okay, this cell is like that specific type. So let's say here in the image on the left, you can see, which is basically a z-score based matrix where x-axis represent different markers and y-axis represent different phenotypes. So let's say for B cell, we can see that, okay, so if the CD20 is high and then the PAX5 is high, then it's a B cell. So pathologists define these phenotypes based on the combination of different markers, right? So now we know that like, okay, if we have the multiplex imaging images, then we with the help of pathologists we can define like different phenotypes. So the problem statement is like, okay, cell phenotyping is important for like tumor microenvironment understanding, and then we have the technology with by using the multiplex imaging, we can do the cell phenotyping. But the only issue is like if we ask the pathologist to manually do, it will be really like challenging and also like a subjective task, because let's say pathologists have to review like 40 markers for one cell. That's too much for a person. So what we really want, we want to like automate this process, and then the obvious solution could be like the machine learning, which can easily mitigate the subjectivity issue and also reduce the labor intensiveness. And actually there are like a couple of methods which they try to address the issue. And there are two type of method. Like some methods use like unsupervised learning, which basically just use the domain knowledge. So let's say they are basically using like, okay, for a specific phenotype, they just focus on what cells we are like what marker we are interested in. Like if those markers have high response, they assign that label, right? So, and then the other type of methods are like supervised method, which obviously use the pathologist like pathologist labels, right? So there, yeah. So now the issue with these methods is like the unsupervised methods are fast, but they're less accurate, obviously, because they're unsupervised. So there is a chance that like we will get a wrong level. And then the supervised methods are like relatively more accurate, but they are like computationally expensive. So what we really want to do, we want to address this problem. So what we did, we like develop a computationally lightweight and highly accurate like cell phenotyping annotation method for multiplex imaging. And then we evaluate the performance of the proposed method using like different data sets and using different evaluation metrics. And then we also compared it with the existing methods to see that like how beneficial is to like propose a new method compared to the existing one, right? So in terms of data sets, we have like used 5 different data sets. So 3 data set was like in-house, which we can say like the private data sets. And then we also have like 2 external data sets. One is publicly available, another one is coming from a collaborator. And out of those 5 data sets, we have like 3 data sets what's coming from the MIBI machine, like maybe imaging technology, and then the other 2 are like from the CODEX. And again, out of those 5 data sets, 3 was from the classical Hodgkin lymphoma, and 1 is colorectal cancer, another one is like diffuse large B-cell lymphoma. One of the reasons that we have like 3 data sets from the classical Hodgkin lymphoma is basically this work is part of a bigger study where we are trying to understand the tumor microenvironment of classical Hodgkin lymphoma, right? So, and then the colorectal one is basically the one we got like the collector one is basically the public one, so we use that to just evaluate our mark. So yeah, okay. So here is a like a brief description, kind of stats of the of these data sets. So each data set have a like different number of markers ranging from 22 to 49, right? And then in terms of phenotypes, there is also like some variation in the number of phenotypes. So basically, it depends like for a particular project how many phenotypes they are interested in. So that's why we got that number. Otherwise, like someone may get a like same number of phenotypes, but it largely depends on the project. So okay, so we got the data. Then the next issue was like the pre-processing. Just like in HNDE images, like we do pre-processing to make sure that like there is no like tissue folding or like blurring. In multiplex imaging, we have like different kind of different type of issues. One of the issues like the channel crosstalk issue, where basically while imaging the section like the tissue sections, sometimes on the machine capture the signal from another marker, right? So here you can see an example where at the bottom left you can see a channel which is kind of contaminated because it has like a response from another channel which is in the middle. So you can see that like okay, there is a pattern, like there are some cells which should not be there. So we use like an existing algorithm to remove this, and then in the clean channel you can see that okay, so the contamination issue is gone, right? So that's kind of standard pre-processing step in multiplex imaging, especially for MIBI images, right? So, and then the next issue is image denoising. So basically, these images also have like noise, and that noise is coming from like various factors. It could be due to the tissue quality or non-specific binding of different antibodies. So basically, I started this project from this step, like where our collaborator asked us to like, okay, can you do something to get rid of this noise? So I was like, okay, what if I just train a like simple foregone background segmentation method, and I use like ResNet uh, to like segment the noise, right? So, and for that, basically, I use one data set, like part of one data set to train the model. And for ground truth, so here you can see an example image. So it's really hard to actually mark a really clean ground truth because there are like the cells are also kind of not in a regular shape. So what we did to generate the ground truth is basically we use like KNN based method to get rid of like all those pixels where we don't really have like much higher intensity in their neighborhoods, right? So with this, and then obviously with some manual correction, we managed to get the ground truth. Then I used that ground truth to train the model, and then I was able to like get the like even better results than the ground truth, because algorithm has learned from the images and learned to ignore all the noise, which KNN method was not able to do. So that's kind of one like really simple thing that I did to get rid of the noise. And then the next step is like segmentation. So usually like in multiplex images, so because the aim is to understand the tumor microenvironment, so that's kind of standard process refers to the cell segmentation to know that okay, how many cells are there, then they do cell phenotyping, like what type of cells are there, and then they do like more downstream tasks to like do like some cell interaction level analysis to further understand the tumor microenvironment. So for cell segmentation, like there is already really like reasonable cell segmentation method which is available on web. So we use that method, which is like deep cell, and this method basically you like um, three are usually two marker. Like one is nuclear marker to know the where is the nucleus of the cell, and then the membrane marker to see the like to get the boundary of the cell. So with these two, it usually perform really well. It again depends largely on the dataset, but in our case we use deep cell to do the cell segmentation. So here is a sample results where you can see that like for the raw image we get like reasonable results. Right. So after cell segmentation, now the next step is like now we have cells, right? And the reason we are developing this method instead of using an existing cell phenotype method is basically the computational efficiency, right? So we decided like instead of working at the image level, we can work at like cell expression level. Like we use the cell mass to extract the cell expression of each cell for each marker. So at the end, for a given image, we get like k cells, uh, uh, and for n marker, like we get a like k by n matrix where the k is the number of cells and n is the number of markers, right? So basically, it really simplify the overall like complexity, like basically in terms of computation complexity, right? Although like there are pros and cons, like doing this, we are also losing the neighborhood information. But at the end, like if this thing works well for cell phenotyping, then this is what we really want. So, and then obviously our method is like a supervised method. So we want ground truth as well. So for in-house datasets, we we acquired the cell phenotype labels through like an iterative clustering process. So where one of the like the team member did the clustering on the cell expression matrix, right? And then they based on the cell expression, they assign the cell types, right? And uh, basically they cluster the cells, and then for each cluster they assign like cell phenotype, right? And then if there are cluster where it's hard to assign a one specific cell phenotype, they do like a further clustering. So this is kind of iterative process where we did like we mark the ground truth with the help of pathologist, uh, and we get like a really large number of annotated cells. So in total for different data sets, so the first data set has like largest number of cells, which is around about 1.6 million. And then for others, the cell number of cells are relatively low, but still like about 100K for each data set, right? So now we have like cell labels, and we have the cell expression matrix. So for the model, like the simplest approach, like one of the aim is to for this like project is like the computational efficiency. So I just use like simple multi-layer perceptron network where I use like four like fully connected layers, and then uh, for the feature vector, I use both like the cell expression metrics, and also I also consider the cell size, so that it basically gives like we know like in some cases, like especially for tumor cells are usually large, and then for immune cells, like there is like different sizes which may help in better cell phenotype. So that's kind of method. So here is the flow diagram where we our input image is like n-plex image where we n markers. We during the pre-processing, we convert this image into a cell expression matrix, and then we feed this cell expression matrix this multi-layer feed-forward network, and we get the cell prediction or cell type predictions. Right. So we train this model for each data set and in a 5-fold cross-validation setting. So where we use 4 folds. We keep 1 fold for testing, and then from the 4 folds we use like 80% cells for training and 20% cell for validation or optimal model selection. So this way, like in all our experiments, like the fold that we have used is not really used for the optimal model selection. It's kind of separate test set. Right. So for performance evaluation, we have used like number of different methods. Oh, sorry, number of different evaluation metrics, including precision recall, FN score, and to understand overall like classification pattern of classification, we use confusion metrics. So yeah, so here is the first result where on the CHL1 dataset, which is the largest dataset we have, and then the overall like average like precision recall and F1 score is pretty high. Like in following slide, I will compare it with the other methods, but it's like reasonable for cell level. Like even in histology, perhaps you know, like current the current state of art for cell classification is also around like 80, because it's really hard to like classify all the cells correctly. So in this case, like pathologist was happy with this performance. So we thought, okay, that's the point to move forward and do the downstream analysis. So, and then when we look at the confusion matrix, it turns out that like the most of the like the mispredictions are usually between the cell types, which are like kind of close to each other, like CD4 T cells and then CD8 T cells, and then in some cases like others which are like obviously the other class may contain so many different types of cells, which we usually like we consider other class just to get rid of like all the cells which with some artifacts. So that's why like this class have some cells from like all the other classes, all the classes, right? So as a next step, then because we have the ground truth, so first we use the ground truth, and then we calculate the like cell expression matrix, right? So which highlight that, okay, for which marker, like for each, let's say for B cells, we have like a high expression of these two, like CD20 and PEX5 markers, right? So we did the same thing for the prediction, and we want to see that like whether this overall cell expression pattern remains same. So if it's same, so which basically mean algorithm is picking the right sort of like the performance is good, right? So and then we also compare our method with one unsupervised method and other like the supervised method, which is cell citer, right? So here you can see the performance of unsupervised method across dataset is quite low compared to the other two, and which makes sense. Like there is a like known gap between supervised and unsupervised classification. So, and then for supervised method like cell citer, like it performed well, but it's still below in all on the data sets. And also like the variation for our method is also quite low. So which is like interesting and also good for us. And then like here is the results, like visual results, you can say. So perhaps it's a bit difficult to interpret, but we have like this last column which basically highlight like which cells are correctly classified with which method. Like we have here two methods. One is R method maps, another one is the other supervised method cell-citer. So if here you see like the pink cells, then it's basically mean these cells are correctly classified only by maps. And if you see green cell, then these cells are correctly classified only by the cell-citer, right? So, and if it's gray, then all the like these cells are correctly classified by both type of methods. But overall, like these images represent that like the overall cell classification quality is reasonable. So we move forward and we come. So yeah. So in my previous results, we observe one thing, like we train the other methods, like we retrain the other methods on our data sets, right? So then obviously one obvious question is like, okay, maybe there is some bias, like maybe we haven't really spent enough time to find the optimal parameters for other methods, right? So for that, like to address these concerns, what we did, we get the results published in one of the paper, which is basically the cell-citer paper, right? So we get those results for the cell-citer, and we get the same data, and then calculate results for our method, right? So as you can see, the results like our method still perform better compared to the cell-citer. But there is another interesting thing, like the results here are basically like the results for the cell-citer are basically an ensemble of 10 cell-citer models trained with a different random initialization. So they said like, okay, they found that like random initialization, like the initialization of the neural network basically matters in their case. So they trained 10 different model, like exact same model, same data set, but just different initialization. So, and they use that, and then they use the ensemble of that to report these results, right? So, which basically mean like if we need to like predict or use 10 model to predict the cell phenotyping, then there is a cost, like the computational cost. So for that, then we did this analysis. So here you can see in terms of computational efficiency, like in terms of runtime, cell-citer is already like taking too much time compared to the other tools, but this time is only for one one model, not for the 10. Right? So just imagine if we have to like use 10 models to predict one, so then it will be like 10 times, right? So that's one of the benefit of our method, is like computationally it's really efficient. And in terms of memory, like the Auster is lightweight and take less memory, or and cell-citer or like take a higher memory or take a little bit higher, but still comparable with the cell-citer. And in terms of GPU memory, it's cell-citer again take lots of memory. Cell-citer is basically a ResNet-based model, which take each image, like each patch, which is I think 64 by 64 patch, and then they use the cell segmentation to get one cell, right? And for each cell, they do the prediction. So let's say if there are like 5,000 cells in an image, they will like generate 5,000 patches out of that, and then they do the cell phenotyping. Right. So, oh, I think there is a question. Okay, that's fine. Right. So now the next thing is like we also want to evaluate the data efficiency, like how many sample do we need to train a reasonable model. So for that, we use a subset of our training set, right? For training set for the CHL1, right? So we use like 5%, 10%, 25, 50, and 75. And here in terms of results, you can see like from 10 to 100%, the difference is not much, but for 5%, there is a slight decrease in the performance, which indicate that like if you have a reasonable number of samples, you will be able to like train a reasonable model for cell phenotyping. So, right. So, okay, our method do have some limitation, which is one of the limitation is like we train model on one data set and then try to evaluate on other then the performance was not that great. But if you look at the performance of other two methods, they also like have like similar performance patterns. So then like this is basically a generalization issue, and it could be due to the lack of standardization between different data sets, and it could also be the overall size of the data. Like if we have a very large data set, like not just in number of cells but also in number of images or tissue samples, then maybe algorithm is well prepared for the like out of distribution samples, right? So that's one of the limitation. And then the other limitation is basically it's kind of generic limitation with this multiplex imaging technology or imaging data sets. One is like they compared to H&E images, they are pretty small. Like in H&E images, we usually have like whole thread images, but here we usually have ROIs, right? And then even to acquire those ROIs, like the technology, overall technology is quite expensive, right? And then also like the storage for these images also require lots of like storage. And like I remember when I was working on the WSI, it looked like, oh, WSI images are like pretty large, which is like around 1 to 2 GB, but here the multiplex images are like around 100 GB. So just imagine if you have a data set with few WSIs, then you have like to you have to have like around 1 TB of space, something like that. And then the one last limitation of this uh, uh, imaging technology is like the lack of standardization. One of the reason is obviously it's new and different people are developing their own technologies, right? So over time we will have like much better like standardization, and we will be able to hopefully like train model on one data set and able to test on another, right? So in terms of future directions, so I think there are like potential future directions. I'm not really sure whether they are practical or not. One of the obvious future direction is like, okay, we know like there are so many limitations with these multiplex imaging data sets. One is like they're small, they are coming from only a small tissue sample, and then why not can like why not try H&E images for cell phenotyping, right? So if we can do that, then it basically means we can do cell phenotyping in so many different tissue types and cancer types, right? And actually there are like a number of paired H&E and multiplex imaging data sets are like available. So if someone is interested, he or she can try those data sets to develop a method. And one of the starting point could be like instead of like training these kind of method that predict like cell phenotyping from H&E images, they maybe like try to start from uh, some H&E based foundation model, like use the features of a patch or a token, and then uh, do the like phenotyping. Maybe instead of like cell level phenotyping, we may consider like patch level or token level phenotyping. Like token is also pretty much pretty small and usually of a cell size, right? So and then the other direction could be like currently like the data set I have used have like reasonably large number of markers. So now the question is like, can we do cell phenotyping using a smaller number of markers? Like maybe there is some hidden relation between these different markers, and instead of using like 40 plus marker, we may use like just 5, 6, 7, something like that. So these are like two future direction that someone may explore, and I don't really have a definite answer like whether like these like there are like 100% chances of success or not. So I have like few links for the pair histology data sets. Like I have like 3 that data set that someone can download from the web. And for multiplex marker prediction, like from like let's say from 7 marker to 10, 20. So there are like couple of existing studies. Not really like they published in relatively low ranked journals. So you may consider it as a starting point for multiplex marker prediction. Right. So that's kind of end exactly 40 minutes. So at the end, I would like to thank all the member of Mahmood Labs and also the member of Cheng Labs, plus all of my clinical collaborators. So here is the relatively old image of our lab, like Mahmood Lab. So yeah, okay, I'm open to questions.
Brilliant. Well, thank you very much, Shaban, for a really interesting talk. It's certainly very informative, and we've got lots of questions. So, shall we open the board questions then? Has anyone in the room or online got any questions? I did see Fias had commented during the talk, sorry, saying that there was very nice work and good to have you at TEA again, Shaban, but sorry, I had to leave early.
Yeah, that's fine.
Brilliant. So yeah, has anyone got any questions? Sorry.
I have a couple of good questions, if I may.
Of course, why not, sir?
Sure.
Yes, first of all, thank you, Shaban, for the seminar. Really nice work. Congratulations.
Thank you.
My questions are, first of all, the first future direction that you showed, that's exactly what I wanted to ask you about. Since you have the cell, you have the paired images, multiplex images, and the H&E, you showed at the beginning that video. Why haven't you tried it yourself? And secondly, what's next with this MAPS work?
Okay, so regarding this self-phenotyping, actually, I have recently working on a larger project, and I have kind of collected multiple publicly available data sets. So all these the data set I have listed here all are public, right? So anyone can use. So currently I'm working on another multiplex imaging based data set, but when I was collecting the data sets, that was my first question, oh, we have both H&E images and multiplex imaging, we can try this, but it's just depend on the time. So currently, because I'm working on something else, so I thought, okay, perhaps someone else can work on this because it's publicly available. So which basically means the competition is really high. So maybe at the same time, 10 different people are working on that. So instead of doing this, what if I just work on something that we have? So it will be then unique and different. So that's one. And then the other thing with the MAPS work, it's basically part of a larger project where we are understanding the tumor microenvironment of classical Hodgkin lymphoma. So basically, they are using this model to do the cell phenotyping in a larger data set. So for in this paper, we use a data set with only few cases, I think 10 to 15, but they have a large number of cases. So we are using the same model on all those images and then doing the spatial analysis to further kind of stratify the patient into low and high risk groups.
Okay, great. And then one further question, if I may.
Sure.
Adam?
Yeah, of course, go for it.
Okay, sure. You also use these 40 channels and then you end up with CD4 positive, CD8 positive, and so on. So you're not really using all of those channels, are you?
Yeah. So actually, in this case, one of the thing is when I started working this thing, we were running against time. They said oh, okay, can you do this thing really quickly? Yeah, I thought okay. So I get all the data and all the channels. I just train the model and end up with these results. But later we figure out that perhaps we only need 20 out of those 40 because out of those 40 there were 20 functional markers, and they don't really know whether those functional markers have anything to do with any direct link with cell phenotyping, right? So then I said okay, because we don't really know whether those extra 20 functional markers have anything to do. So let give this thing as an input, and then algorithm will decide. So that's why, because I have the data with 40 markers, so I use all 40. But hardly for these type of cell phenotypes, we can only use these markers, which are like around 20. So yeah, so that's basically also the question, like, what if, can we do the same thing? Like, can we do the same type of cell phenotyping with way less markers? Because there is another imaging technology which only supports 7, but it's also cheap.
Thank you, Shaban.
Brilliant, thank you. So my next question really is: so for some of us naive computer scientists, what's the importance and what's the use case, I suppose, of being able to do this phenotyping? How does it help us to better understand disease? Or
okay, yeah. So it basically, like, let me go to over here. So yeah. So let's say if you only know that, like, in a given tumor, like here in this case, like in H&E, like when I was working on the H&E, usually I was able to like figure out that, okay, which part is like tumor and which part is normal, right? And but obviously pathologists can do way more than this, right? But there is still a limit, right? But if we know, like, these markers are basically protein level markers, right? So they are representing protein expression. So with these markers, we have way more like detailed information. Let's say a cell that may looks like lymphocyte on an H&E image, in multiplex image, it could be a like a specific type of lymphocyte, and there are like some subtypes which are like good for patient prognosis, but some other subtypes are bad, right? So apparently at H&E, a cell is like lymphocyte, but we don't really know like whether this particular lymphocyte is good for patient prognosis or bad, right? But with these technologies, we have way more information, and then we can figure out that like, okay, there are like subtypes, and this subtype is good, this subtype is bad, and then obviously we can get a like better, we can develop better drugs for that, and also we can stratify patient and then have a better treatment plan for the patient. So in that sense, it's really useful to really understand the tumor microenvironment.
brilliant, thank you. And also in terms of, so of course at the moment that you said the data sets are relatively small using this sort of spatial proteomics. What's the how sort of cost prohibitive is it? Is it, so compared to say a standard, just taking a standard H&E slide and staining it, what's the sort of, is it thousands of times more expensive?
Yeah, actually, yeah. So usually I don't really know the exact cost, but basically it's like if you are developing something of WSI level, maybe with 500, 600 WSI, we can only do like ROIs, perhaps only like 100 or 200 ROIs maybe with the same cards. It's kind of like that expensive, and especially it also depends on the antibodies, how many antibodies we want to use. Like if we are using like low number of markers, then obviously it will be a little bit cheaper. But and then the other thing is, because this thing is only for like tumor microenvironment understanding, right? So we don't really need like the whole like the whole tissue. Like we like pathologists just pick a like a region of interest which makes sense for them, and then we only do like multiplexing for that. So it's like this is how they basically save the cost, and they don't really go for WSI level multiplexing.
Thank you. We've got a couple of questions online as well. So Brenda's asked, would you consider linking the data you have with the histology report? So the diagnosis of some lymphomas is dependent on only a few key stains. So I can't read that, particularly for CHL, which rests on the presence of RS cells, where positive and negative are both very important. Brenda, I don't know if you want to ask that question again. It's quite a long question, sorry, whether you can.
I am incredibly sorry for my fast typing, and no, it was the maps were interesting, particularly from the perspective of classical Hodgkin lymphoma, because I know you're looking at the spatial aspect of the other cells that are there. The technique is useful, I suppose, but obviously it would be good to know for my purposes as to how useful the technology is diagnostically if from the off, and realistically, all of the classical Hodgkins are only really being called that because of what I think you labeled as tumor cell in your data in the little table of expression.
Yeah, I think we have like tumor cells, which I think basically represent the classical Hodgkin lymphoma. There was one.
Yeah, so the tumor cell at the bottom, that is the RS cells, presumably.
Okay, so I don't really know too much about like the exact like phenotypes. So I'm just like relying on the pathologist. But yeah, so in terms of their utility, so basically this method is mainly for you can say the tumor microenvironment understanding. It's not for like the diagnostic, right? So basically the aim of the project is to like understand, like let's say after this project, if they manage to figure out that like, okay, there is a subset of patients and they have like that kind of tumor microenvironment and they have a like good prognosis, and then there is another set of patients, they have that kind of a tumor microenvironment and that's bad, right? So which basically means now they get the knowledge, right? So now they like in the diagnostic, they will just need to perhaps they don't really need to do the exact same number of markers. Maybe they just do like some ISE based stains and then only do those markers which are interesting. So here they are like using a really large number of markers to just to figure out that like which marker is useful or which phenotype is useful. Let's say if we manage to find like the two groups of high risk and low risk patients just based on B cells, right? So which basically means for the future patients, we don't really need all other phenotypes. We can only use these two markers and then decide that like, okay, what's the cell population and whether this patient should give a like treatment based on like the poor prognosis or the good prognosis.
Yeah, does average cell marker expression matrix would that miss out? Forgive me if it doesn't make sense as a question, but would that miss out significant staining of a rare cell? So like take PAX5 for instance, that's
Right.
like, you know, that's a really, really important stain in classical Hodgkin lymphoma because your RS cells are diagnosed really when there's very weak expression of PAX5. But if your
Right.
tumor cells are RS cells, is the very weak staining that you've seen clinically, that's actually incredibly useful. But here it looks negative. So it's not so is it really not just you're focusing on the very deepest stains? There could be important negatives here as well, I suppose.
Okay, yeah, I remember when I was reading the description of this project, and this was one of the things, like there are like really, really few cells which they are interested in, like few cells in term of numbers, right? So if we are doing some sort of averaging, so but anyways, like this Z-score is just for kind of visualization. Like we are not using this thing for like classification. We are using the cell expression feature of each individual cells for training, and this is just for like the visualization, and it may have some impact on the ground truth, but because the pathologists are actually interested in this disease, so I believe they have make sure that like for these specific marker, like they classify all those cells correctly.
Oh, no, cool. Thank you.
So like while like annotation, they do use like some similar strategy to basically like all the different phenotypes, but because they are particularly interested in like classical Hodgkin lymphoma, so I believe they like give more attention to this marker while annotation.
And just out of interest, does your data set include patient response and like outcome data for the patients?
So in this work, we don't really need, because it's just like the phenotyping, but in a larger like in the larger project, we do have those labels, and that's basically what we want to like. Based on those labels, basically we want to understand the tumor microenvironment. Like we want to like classify those patients into good and bad, or a good responder or a poor responder of a particular therapy. So
Okay.
yeah, we do have those labels.
Oh, that's fascinating. Thank you very much.
Brilliant then. So, well, do we have any more final questions from anyone online or in person? If not, I suppose I'll finish with one final question. So I've not used spatial proteomic well, done any form of spatial proteomics before. I suppose quite a lot of our lab probably haven't either. So just can you briefly explain some of the challenges that you've faced going from saying, well, in using this sort of analysis and doing this sort of analysis? Sorry.
So challenges in this technology first is for me was like the domain shift, because I was like used to work in multiplex, in the H&E images, and I know like what type of pre-processing I need to do, right? So even just looking at the images, I can tell that, okay, like, oh, this image has that kind of artifact, so we need to do something. But in this case, when I started working on these images, it was like, okay, I don't really know like whether this particular cell is it something useful or it's an artifact, right? Like just like the very first thing, like the channel crosstalk thing. So just imagine if I just present this thing to you as a marker, right? So I don't really know like whether this cell is like basically an artifact, right? So that was one of the challenge. So basically then we collaborated with another lab which basically do multiplex imaging all the time, and they have the domain knowledge. So they helped me in that sense, right? So I was helping them in terms of like the computational method, and they were helping in terms of the pre-processing and overall domain knowledge. So that's one of the challenge. And then the other challenge is basically the generalization, like whatever you have developed. So it's like it don't really feel good when you try the same thing on another sort of data set, and then you get a like relatively bad results. So ideally, like we want to develop method which we should work for other data sets as well. So I think over time, once we have like enough samples, then we will be able to like train model which will work across different data set and across imaging technologies. So yeah. So
Brilliant.
if we don't really have any other question, I have one question. Like, is anyone in like TIA Center working on multiplex imaging? I think Sean, I read something on his profile, like he's interested in something, but I'm not really sure.
I think there's a few people are looking into various.
Okay, that's good. Hopefully we will get more data sets then.
Yeah, definitely. Sorry, Banaz. Okay, brilliant. Yeah, of course.
Okay.
So yeah, Banaz is in our lab is working on multiplex imaging. So
Okay, good. That's good to know. Okay, then thank you for inviting me.
no, well, thank you very much. Then we'll call the seminar to a close then. But thanks a lot, Shaban. It's been really interesting, and it's been good to see you. So and best of luck with your future work.
Thank you.
Just before we close it, I suppose just wanted to remind everyone that our next seminar will be next Monday, so the 15th, same time. We're joined online by Professor Hamid Zouch from Mayo Clinic in the US. So hope to see everyone there. And again, a final reminder, so for the MIUA special session next week is the deadline, so the 15th as well for the full papers. But thanks again, everyone, and thank you very much, Shaban.
Okay, bye.
Bye, everyone.